Good morning and welcome to Biology 102 Tea Time. I've grabbed a glass of tea. I highly recommend a little bit of caffeination to get you through this. And if you're wondering if I'm wearing pajamas, yes, I am. And I think that you should be too. So today I'm going to walk you through the cell cycle and DNA replication. If you missed our lecture before spring break, we covered the chemistry of DNA. So go back and review that. I'm not going to do a video clip of it. But today I want to sort of walk you through two things. One is the cell cycle. This is the phases that a cell goes through during its lifetime. This can be before, during, and after cell division. So all of that is included in the cell cycle. And I have this arrow over here. I'm going to walk you through G1 phase, S, G2, and M, and just kind of talk briefly about what happens in each phase. So G1 phase is it's known as gap 1, but it is the place where the cell does most of its normal function. So if you're a red blood cell, you'd be carrying around oxygen. If you're a pancreatic cell, you would be producing insulin or something like that for the body. At the end of G1, a cell will decide chemically whether it wants to divide or not. This is determined by whether there's nutrients present, whether there are sort of a big enough cell or there's enough cytoplasm and organelles inside, whether it's needed inside the body. Why divide yourself and waste all that energy if you're not even gonna need an extra cell? And so the cell will decide chemically whether it wants to divide. If the cell decides to go through cell division, then it will enter S phase. S is for synthesis, so that means you're making something, and in this case you are making more DNA. You are going to copy all of your chromosomes through a process called DNA replication. In S phase, for a human cell, you would start with 46 chromosomes at the beginning of S phase. That's in a normal sort of pancreatic cell or a normal sort of body cell. And by the end of DNA replication, you will have 92 potential chromosomes in that cell. And so this is the phase where we're going to copy our chromosomes. G2 is all about prepping for cell division. So you will divide your centrioles. If you're an animal cell, you will get ready to sort of divide the cell. So make sure you have enough mitochondria. You've also just copied all these chromosomes, and so you might want to check to make sure that you haven't made mistakes, and so you'll proofread your chromosomes and make sure that they are ready to go. And then this last little piece of the pie is called cell division. So this is where we will be making new cells. Now notice up here, right here, it says interphase. So in high school, you might have learned G1, S, and G2. Those three phases are collectively sometimes called interphase. So I want to dive into S phase and talk about what happens with DNA replication. DNA replication is the process where you're going to copy all of your chromosomes. This is important because you don't want to make new cells and then have nothing to put in them, right? That doesn't make sense. And so S is for synthesis. At the end of this phase for a human cell, if you will look at over here, you start with 46 chromosomes in a human cell, and you will have 92 potential chromosomes at the end of DNA replication. Chromosomes are often shown like this, like these letter X's almost, and this is showing one chromosome and then its identical copy. So that's often why we're showing a set of two, is because DNA replication has already happened. All right, there are many enzymes that guide DNA replication, but I want to walk you through 5. And so this will not be all the enzymes that are possible and all the enzymes that help. If you watch other videos, you might see some other enzymes, but I do want to walk you through these 5 to tell you a story about how DNA replication occurs. And I'll be using these chromosomes down here at the bottom to kind of show you what is happening. So the first enzyme that I want to mention is helicase. Helicase is an enzyme that is named for the helix, and it unzips DNA, we say, or the two sides of DNA will be pulled apart by helicase. Then once the DNA strands are separated, primase can add a short primer. If I look down here, helicase has already separated our two pieces in letter B, and so primase would jump on and add a RNA not DNA, but RNA primer to each of the ends of DNA, and it will be the opposite ends of DNA. Then it will add a short sort of letter section of RNA, and that is necessary because our big copying enzyme, kind of the Mac Daddy copier of DNA replication, is DNA polymerase 3. And DNA polymerase 3 cannot get onto a single strand, so we need that primer to make a short double strand. It's almost like you're braiding hair, and you need that knot at the top of like sort of the piece of hair to then braid from. The same thing is happening with DNA polymerase 3. It can then attach itself, and then it will copy a new strand of DNA from the 5' to the 3' direction. My fourth enzyme on my list up here is DNA polymerase 1. DNA polymerase 1 then comes along and cuts out the RNA primers, because you cannot leave RNA in your DNA strand. It has the wrong sugar, it has uracils, that's never going to work. So DNA polymerase 1 will cut out your RNA primers and replace them with DNA language. So we'll replace those primers with DNA language. However, the DNA pieces are now not attached. There will be gaps between them. And so our last enzyme, DNA ligase, comes along and seals up all of those gaps so that you have one continuous piece of DNA that is then flipped into a double helix. So these are the 5 enzymes. There is a simulation you'll be doing in lab. We will also be drawing this out on Monday, 3/23, so you can take a look at how that looks. And there are some recommended animations that you can look at that will help you visualize this process. Thanks for joining me today, and I will see you next time.